Very good. Okay. So thanks, Grant, for the excellent introduction to the field. Really, as we are here now partly to learn the new frontier of the field, but also partly celebrate what cryo-EM has been able to do. Right? Started this actually it's been built up for decades, but so-called cryo-EM resolution revolution started more than 10 years ago. And we can see the power by the past 2 days already. You see the single particle type of work can bring really very high resolution. And today you heard from Grant to talk about really the next frontier. Really, what we want to do is actually we're thinking it's a revolution, but a lot of us think the revolution is actually yet just about to come. That's when we can do similar things in situ. In situ, as Grant already said, is basically inside the native environment of the species. Obviously, cell is one of the bigger goal we have to try to conquer. Then, of course, for these in situ samples, there are a lot of challenges you have already heard. There are first actually sample prep. Unlike your purified samples, need special preparation because the cell sample, for example, very thick. You have to mill it. Then even if you manage to get a very good specimen, the nice thickness, good contrast we can see, still there's very high background there because everything is inside the cell. And also there are all the molecules. But on the other hand, you can think this could be a challenge how to deal with this, but also it's a blessing if you could deal with it, meaning when you collect data, everything's in there in your data set. But on the other hand, of course, because of the high background and the crowdedness, then it's going to be hard to achieve the same, say, resolution to the same size of molecule. Basically, we have a bigger size limit. Those are the challenge the field is still facing. Then on the similar line is actually, other than background, also there's the intrinsic heterogeneity and the dynamics of all the samples. The whole pathway of your molecule of interest is in front of you. Again, very hard to tease out, but again, if you could do it, it's actually you learn so much more. And then finally, not finally, it's actually just one more example is actually your target may be very sparse. It's not purified, it's not enriched. So there's ways you have to deal with the abundance challenge of your target. But those are things on the frontier of development. We're getting inching closer to get this figured out. So as Grant already mentioned to you, you learned the single particle cryo-EM in the past couple of days, and today you start hearing the great power of cryo-ET. What I'm going to actually introduce you today is actually a method sort of in between the two. Really, is actually we're going to use single particle cryo-EM to deal with in situ samples. So hopefully we can actually bridge the gap between the two. So I'm going to give you 3 examples how we do this, sort of almost like a hierarchical to build up the complexity. Start with some viruses, meaning we can say viruses is a native virus particle, still a native environment. We still call that in situ, but the size is bigger than what your purified sample normally is, but still manageable by single particle. Then we'll go on to purified organisms like mitochondria. Then finally, of course, we want to go into the cell, go into the female thin sliced cell. So 3 group of examples I'm going to go through. So first, let's start with virus examples. We traditionally study HIV. One of the viruses, of course, is a pandemic causing virus. Then the challenge of this is actually for HIV, it's not your typical type of very highly symmetrical, like flu type of particles. You have this very nice icosahedral symmetry. You can do traditional type of single particle. For HIV, it's actually in illustrations, you see nice symmetrical particles. For example, this is actually just a little figure showing the maturation of HIV virus. It's actually take 2 different shapes. In the immature form, it's almost spherical, but it's broken. It's not good symmetry. Then in the mature version, you see the conical shaped virus that you see it everywhere. Again, none of these are real symmetry in there. meaning it's actually overall it doesn't have a good symmetry. So you cannot just treat the whole virus particle as a single particle. So traditionally, it cannot be done in a single particle way. Then how it was done is really, again, through cryo-ET, cryo-electron tomography. So this is almost, it's a landmark work by in John Brake's group, really sort of almost pronounced the arrival of high resolution cryo-ET. That's done almost 10 years ago. And John's group got this down to 3.9 angstrom. Really, you can build side chains in atomic models into this type of density. Okay, then this is really the why cryo-ET has to be done. Is actually this is a work shown in Peijun Zhang's lab. Really highlight the issue of this. On the left, C and E, you see that those are the sort of the capsid mapped into the data. This illustration, you can see that although they all look like spherical, but they have different sizes and they're broken. So that's why you have to deal with each particle individually. You cannot say the two particles are the same. You cannot average them. You can only do sub-region with sub-tomogram averaging. But that's cryo-ET, right? Then the field gradually realized, although these particles overall, they don't have a conserved big symmetry, but locally, say the capsid protein units, how they interact with each other locally, they still obey local symmetry. Then what if we treat local symmetry as a local single particle, then do the single particle way? So that's what happened. The groups in Owen Pernillo's group and the Rob Dick's group just a couple of years ago started doing this, and they're very effective. On the very left, you can see those white circles are just basically treat the sort of a region of the capsid as a single particle, just a local small region, then pick those regions, just like you do your purified sample. In that case, we average locally, you can get pretty high resolution, saying the two studies are by the same approach. So I will introduce a little bit more on this. So we did this among other groups, look at the immature capsid. Again, you can see this on the micrograph. Again, overall spherical, but they're never the same. They're all broken. Okay, this work, the people in my lab done the work. I just want to acknowledge them there. So again, we can pick single particles in the local region of these capsids, then average them, work very well. Okay, this is the 2D class averages of the picked single particle. You can see the nice net lattice pattern. Then we just use standard, say, single particle cryo-EM reconstruction tools, as in CryoSpark, get to quite high resolution. Okay, I will introduce you a few applications of this method. Also, sort of draw your attention to the sort of a view I will show you. On the top is a larger region, the patch of the capsid. On the bottom, it's almost like a hexameric building blocks of this on the top view and the side view. Okay, so these are the overall the characters of the capsid. So we want to study this. Okay, so it's sort of a going in situ, meaning in the native virus environment, using this virus-like particles, VLPs. Virus-like really means we did the mutation to the virus, say, get rid of the virus envelope or spike. So it's not infectious. We can handle it in our BSL-2 microscope. So you can see that on the right are some micrographs of the viruses. You can see the nice lipid bilayer. Then underneath is actually you sort of some regular pattern. That is the capsid lattice. So we did this with this, and again, very high resolution. We can see almost all of the side chains and also the small molecules that actually the virus soak up from the cell to stabilize the capsid, the IP6 cofactor from actually our cell, human cell. So all seen very well. Then take this step further. We would like to do, say, inhibitor or drug design to these viruses using cryo-EM. And then to facilitate drug to get into the virus, we actually take advantage of a system of bacterial toxin, the PFO, which is known to poke holes on the lipid membrane, cholesterol-rich lipid membranes, which is actually the HIV envelope has. So this has been done before, actually, looking at the mature HIV virus by Theo Barkin's group for imaging studies and Pei-Jun Zhang's group for cryo-EM studies to say basically increase the contrast or let small molecules to diffuse in and out the lipid envelope. So these are done in mature virus. The capsid is separated from lipid envelope. We would like to ask for the immature virus, the capsid is actually lining the lipid envelope. We want to see whether this method still works or not. So we started doing that, worked pretty well. So all this is on the left is the intact virus. On the right is treated with the toxin PFO. You can see the holes actually form on the lipid envelope, and you can still see the lattice of the capsid mostly maintained. So it works. So really the advantage of this is really we can study inhibitors very easily to actually get in and out of the lipid envelope. Even if our goal, if you want to do drug design, it should cross the lipid membrane barrier, but you use this method, you can study the compound easier even before it's optimized for, say, for membrane permeability. Okay, so we started using this to study a drug inhibitor compound called Biravimab or BVM, which is actually known in the 90s has inhibited HIV replication. Then only in the 2018 was the first structure of the BVM bound to the HIV capsid being determined in Mark Yeager's group, actually using micro-ED, the electron diffraction method. But the resolution wasn't too high there. As you can see in these figures published in the paper, the density of the drug is not too good. It's actually barely fit of the compound. Then a couple years ago, the Tatiana Poliano-Nova's group at University of Delaware had used NMR to look at this structure again. Then also come up with a fitting of how the compound binds to the HIV capsid. But the issue is that if you superimpose the two models, from NMR and micro-ED, you see they don't match. So still, it's an open question where the drug binds. So we, of course, using our in-situ cryo-EM to look at this to get high resolution in the native environment, hopefully resolve this issue. So in this case, we get to very high resolution. It's about 2.1 angstrom resolution. That, again, showing the two views that we can say on the top view that you can see here, the IP6 molecule, very important for the virus, is the host small molecule cofactor. Then just underneath the IP6, we can see Biravimab, quite very nice density. It looks like a tube, even at this high resolution, because it sits on the symmetry axis. So it's symmetrically average, 6-fold average. But nonetheless, we can fit the BVM very nicely into this contour. So when we compare to the previous study, it turns out it doesn't look like any of them, or the location-wise, meaning it's actually shifted from both studies. But we have higher confidence since we did this in the native virus, and also our density is actually very good. But to confirm this, we teamed up with our collaborator Juan Perea at the University of Delaware to do full atom molecular dynamics simulation to look at where the BVM binds. What Juan did is actually just take this molecule here, let it glide through the central pore, along this pore, and see where is the energy minimum for the drug to bind. Then calculate the energy minimum, then pick that position and the drug conformation, then compared with our cryo-EM study. And lo and behold, if you see here, it's actually matched perfectly with our observation. So now we have a better confidence where this drug really binds. Okay, so then moving along, we look at another important drug molecule, lenacapavir, which is actually the breakthrough of the year for science last year. Really, this is a small molecule inhibitor, once per 6 months dose. You can actually administer to the patient, really long lasting. Why it's long lasting? Because it binds to the HIV capsid in picomolar affinity. So it stays there to inhibit the virus almost forever before the drug is degraded. So it is known to target the mature virus capsid. Okay, but also in the virology study, also known that actually lenacapavir not just targeting the sort of the mature form of the virus, somehow it also inhibits maturation before it gets to the mature capsid. So there's some hint that this drug also binds to the immature virus, that there's no biochemical or structural data available at the time. So we looked into this using our system. Again, we captured the structure of it. This is actually the same data set. We treated both BVM and the lenacapavir to the VLP, the PFO perforated VLP. What we see lenacapavir density very clear. Again, just orient you earlier. I showed you down there is where BVM or Burimet binds. That's the IP6. Then lenacapavir binds to the N-terminal domain of the capsid. Actually, in this view, it's up there. So very good density. In this case, it's not on the symmetry axis. It's not average. Then we can fit all the functional groups. Really, I don't want to get into details. Just give you some impression or idea that in situ structural biology is very important for studying virus. Okay, very effective as well. Okay, but that's not really what I want to focus in from the sort of the technology point of view. Let's move on to the next level. Okay, want to do, say, in situ cryo-EM of purified organelles. Again, it's not purified sample. It's a piece of the cell, contains everything. Okay, I want to highlight this with a study from my colleague Kai Zhang's work at Yale. It was published last year. So Kai's lab did is this. They took the pig heart, then purified mitochondria from pig heart, then did both single particle cryo-EM and cryo-ET on it. Combine the two, but mostly rely on the single particle cryo-EM data to get to very high resolution in the respiratory super complex chain. So really it gets very high resolution, about 2.5 angstrom resolution. That is in situ, it's not purified complexes, but you can see this very nice type of nice densities. Now, what you can normally you can do with purified sample, you can do with this. Just build high resolution atomic models and also fit all the ligands. Then the beauty of this type of work in situ is this. It's not the condition is minimally perturbed. We should not say it's not perturbed at all. Almost as much is inside the cell, so that you don't lose some transient state. You don't lose component that is actually weakly bound during purifying. You may lose them, right? So in this way, Kai's lab identified many different conformations or complexes of this respiratory chain super complex. So that's actually not been observed before, and that could actually come up with a better model how the electron transfer occurs during the respiration. So again, not too much biology, just want to show you the power of this method. Then in our group, we also look at a different complex from purified. In this case, it's actually human mitochondria. We purified it and look at the mitochondrial ribosome get to pretty high resolution. Then you can see it's actually very nice. Of course, the final reconstruction is that this resolution looks really nice. You can see many factors bound to the mitochondrial ribosome. You can see the nascent peptide chain from the coming out of the channel, the tRNA there, and also that goes into the translocase that actually pumped the peptide out of the mitochondria in this case. Then one thing again about doing the structural biology in situ is actually you don't just look at static structures. You look at the full spectrum as it happens in a living cell, right? So we can actually see a slew of different conformation or different translation states of how mitochondria work. And we did this in a wild type cell and also in a factor knocked out cell. We can see how the actually particular factor actually regulates the translation landscape or the translation cycle. So again, so this is the power of the in situ structural biology. Okay, I quickly glossed over this. Also, just want to show you the concept in situ structural studies with purified organelles actually can be very effective for the particular problems you're looking at. I want to spend most of the time talk about the most enticing sort of target we want, right? For many of us for decades, is really can we do the type of structural biology that we do with purified sample in the test tube directly in cells. Okay, so this is really what you heard from Grant. Also, where really spearheaded this field is the development in this field is spearheaded by the development in cryo-ET. As you see, that start with sample prep. Cell sample is very thick to be in if we want to image it with electron microscope, we have to thin it, right? As you see, the cryo-fit milling is one very popular and effective way to cut the cell into very thin lamella, then use tomography way to reconstruct the three-dimensional structure. So Grant already showed you this animation, so I can skip. Now it's really the tomography. Actually, Grant didn't show you the high resolution type of tomography. It's actually very impressive, very amazing. This actually very high resolution, atomic level resolution. Atomic meaning you can build atomic models in the low 3 angstrom type of resolution already being achieved for large molecular complexes like ribosomes in a variety of cells after fit milling using cryo-ET, mostly done in Julian Muhammad and Martin Beck's lab. It's quite stunning work. Okay, that's the power of cryo-ET, as you already heard. But also, as Grant alluded to, or at the end summarize, cryo-ET is extremely powerful, but also extremely hard to do. There's some high tilt intrinsic problems that have to be dealt with to get high data quality. The field is very actively working on that, but we don't have a good solution yet. And the whole process is very tedious and they require high level of expertise, which actually not many people like Grant can do it. We say, we're just not as good. What can we do? So some of us thought, can we take a shortcut? Really basically combine the fit milled cells with single particle cryo-EM, as I showed you earlier with the native virus particle with purified organelle. Can we actually do this directly in cells, directly on fit milled cells, traditionally apply cryo-ET to it? So this is how we actually started doing this work, but this idea is not really unique to us. Many people thought of this, a few groups really very seriously done the similar type of work as well. Most notably, Xinjing Jiang's group at the Biophysics Institute in China and the Nico Gregorio's lab at University of Massachusetts. So they actually get quite nice result already sub 3 angstrom resolution with the large molecular complexes. Then also along the way, both groups, Xinjing Jiang's group and Nico Gregorio's group, actually, they also wrote software to help the process. Basically, the question is, how can we identify and align the single particles in a very noisy 2D micrographs? These are not tomograms. That's Grant shows. The signal to noise is actually even way worse than that case. So there are ways to deal with it. I'll show you our pipeline of how we do this. So our work started about 2 years ago with a fantastic postdoc, Wei Zheng, showing picture here, who really spearheaded the whole process, push it all the way to the end. So show you some data. On the left is basically frozen cells on the cryo-EM grid. Those bumps are frozen cells. Those marks really marks the position we wanted to or designed to mill, to do the fit mill. in the middle, you see the typical lamellae that actually thickness about, in this case, for single particle study, we want to mill it a little thinner than the cryo-ET type of study because we have actually less signal to noise in 2D than 3D. So we target to about 100 to 200 nanometer thickness. So film milling is a bottleneck to this work. We received a lot of help, assistance from our colleague. The Jian Feng is actually our facility manager at Yale, and also our colleague Jun Liu really helped us a lot to get this going. So for today's work, I'll talk about we basically the two types of experiments I will talk to you about. For native cells, we milled about 200 lamellae, about 12 days, and inhibitors treated cells about 100 lamellae from 6 days of milling. Okay, really this is the bottleneck. It actually takes months to train us. Actually, Jun helped us. We all allowed up to Jun Liu to really make this happen to us. Now we actually get much better than this. We could actually efficiency improves about, we could do maybe twice as fast, maybe 3 times as fast these days. But still, this is still something the field or the instrument, the commercial available instrument need to develop better so that become a common practice for the field to take up. So after this bottleneck, the work become actually quite more or less standard or very enjoyable, as I will show you. Okay, so after, I think someone marked on the slide if you can clear it, but if not, it's not a big deal. This is actually after this is a typical lamella we milled. Then we worked with our colleague Kai Jiang and Shenping Wu to develop a very efficient single particle data collection scheme on all the milled lamella. So here's the low mag view of the lamella. Okay, so what we can do is just like your purified sample, just like your purified EM grid, we can say, this is the location we want to collect data. Let's focus on this location. We can just do zero tilt, multi-shot, shift the beam to a different location to collect single particle shots. Then do the same thing, move to all the different positions to the entire lamellae so that we can get very efficient data collection. These are typical micrographs we got out of this. And this is actually very efficient, almost like your purified sample. We can get 4,000 to 5,000 micrographs per day, okay, for single particle micrographs per day for data collection. Okay, and then these are the sort of a gallery, small gallery of different micrographs of different parts of the cell. As you can see, down in the middle is the piece of mitochondria. Then upright is actually nuclear pore complex. And on the bottom left, you can see lots of microtubules, all the cell features. But we focused our first study on, so if you look at the top left, the 2 micrographs, we focused on those black dots, which are, I think, perhaps the lowest hanging fruits inside the cell. Those are the ribosomes. So, but this is the very first data set that we collected on the single particle milled cell. We don't know how this will go. Then we were discussing during data collection, we were saying, if the first time we do this, then if we actually get a resolution in the final reconstruction better than 10 angstrom, this should be we can call this a successful experiment. But it turns out actually the results are way better than we imagined. This is actually an email sent out to the team second day after data collection. Using everything just goes through the CryoSpark standard pipeline. We get 3.6 angstrom resolution 1 day after data collection. Okay, then really get excited about this result. We started to assemble a large team, try to optimize the process, computational process. Then we also take advantage of using the 2D template matching, mostly using the software, both of the software GSPAR written by Xinzhen Jiang at the Institute of Biophysics in China, and also the 2D TM by Nico Gregory's group. So what we did is this. I'll go through a little bit since this is not entirely standard, but more or less all the tools are available for the single particle cryo-EM, but I'll walk you through the procedure we did this. So this method depends on a reference, meaning available template, a prior reconstruction. You have some information already, can be at a very low resolution, but you need a starting reference. That could be a problem, right? Those in the field long enough know model bias is always a concern, that if you don't see things clearly, then you can interpret your result or bias against your reference very easily. So what we did is actually we're looking at a ribosome. It has a large subunit and small subunit, makes the 80S particle. So in our reference, we took only the 60S, the large subunit, without the 40S small subunit. Okay, so we identified a lot of particles with this template matching method, about 3 million to start. Then we started to do classification. Then what we do classification is actually we create a mask focusing on the small subunit only, which is never, the data never seen it, or the particles, really it's not in the reference. Okay. Then we look at the appearance of the true ADS particle. If they show up, those are not model biased, not reference bias, right? Because the 40S information is never shown in the template matching process. So with this, we're confident that we're getting the true particles. Then in the end, do many cycles of refinement and classification, we can get a subset to very high resolution. In this case, it's about 2.5. Now we can actually do focused refinement to get the large subunit, small subunit, because some of you, if you know the field, the small subunit actually has a head and body domain, they move. So we can separate them, focus, refine to high resolution. Then for the rest, these are subset of particles. We have another set of particles is actually match our reference. There we need to be even more careful. So there are multiple ways. We could actually almost like a omit map. For those who does crystallography, you remove part of your reference, part of your model. So in this case, we removed a large RNA helix from the model. Then we see in the final reconstruction, if it comes back, we know it's correct. And also, all of our searches started at 8 angstrom resolution. If we get to much higher resolution, and also makes sense, that as RNA should look like, protein should look like, we know it's not biased. So in this way, we can get the bona fide 60S particles as well to quite high resolution. So show you some results. That's a month later, we can get 2.5 angstrom resolution, pretty good density. Then keep working on the method or classification and particle picking. A few more months, we get very high resolution, better than 2.2 angstrom resolution, and the local resolution is actually better than 2. Actually, we collected data, the Nyquist is about 2.1. So this is actually better than Nyquist since we use a super resolution mode. If we use actually smaller pixel number, we could actually get even higher resolution. But anyway, it's good enough, really stunning the type of cryo-EM density. We can see lots of holes in the aromatic side chains and nucleotides, and also lots of, even for the small amino acid side chains, we see very good definition. And we see a lot of post-transcriptional RNA modifications like this methyl group on the sugar and the methyl modification on the bases. And also we can tell this is a pseudouridine. It's not a cytosine because it's actually the water is here, hydrogen bonding so to this position. So it cannot be hard carbon, it has to be a nitrogen. So we know it's pseudouridine. So really very high quality. We can do a lot of analysis on this. As I mentioned earlier, that we treated the cell with the drugs. We actually treated the cell with in another data set we treat this cell with the most common ribosome inhibitor, the protein translation inhibitor, cyclohexamide, and also one of the, it's actually the only FDA-approved anti-tumor drug targeting human ribosome. That's called homohydrotonin. So we treated that, and we see very clear, very nice definition of the drug density as well. So you potentially down the road, people can do drug design directly in cells at high resolution. Okay, so lots of good, not just this, right? We can look at the interaction with the codon, anti-codon pairs between tRNA and the translating mRNA. Very nice definition of density. But for people who know the field, or for most of you, if I give you some time to think, you may ask, actually, in the cell, everything's going on, right? For protein translation, there are about almost 60 plus tRNAs, bring more than 20 amino acids to at least 4 different sites on the ribosome, and ribosome is actually swiveling and ratcheting, lots of conformational change during this process. Basically, once you do high resolution work, you do any type of structural biology work in situ, basically we have this situation, everything everywhere all at once. So as I said early on, this is a challenge to all of us. Can we distinguish the different translation states? For example, in this case, this many different states in the data set. But again, if we could do that, then it's a blessing that we learn so much more of the process. So we applied the so-called purification in silico, which is basically just 3D classification of the different particles we got. And I'll walk you through the process. We first look at put again 40S mask, look at the rotation of the 40S domain relative to the 60S. So we can classify the particle into different rotation states between the large and small subunits. Then we focus on the factor. Factors means if you focus on the middle graph, it's really showing the different type of tRNA and the translation elongation factors binds on the ribosome. So in the end, we got 23 different classes out of this. All of them, apart from one at about 3.3 angstrom, all the rest is actually better than 3 angstrom resolution. So very high resolution. And also we can count precisely the particle numbers. Since the particle quality is very high, we can do statistics. Basically, we can know what is the abundance or the translation landscape of different classes or different translation states inside the cell. So just show you an animation that we start showing a hibernating ribosome stage, no mRNA, no tRNA. Now in a different class, we can see the P side tRNA comes in, another class, AT tRNA brought by the translation factor EF1, then goes to the A side, bump to the E side, and more and more and more. In the end, we see many of them, the entire cycle. Okay, these are for the translating ones that we can see how really a very comprehensive picture, how the tRNA basically dance through the ribosome for the complete cycle of translation. Then, of course, we also see the non-translation states as well. So the biology-wise, perhaps I'll give you one example. What we learned the most interesting is from this molecule called SIRBP1. This molecule is known in the ribosome field. It's called a hibernating factor because from purified sample, studies from purified sample, it's known this shaded region. It's a very long, almost intrinsically disorder or noodle-y type of molecule. It's not folded. Then the middle part, the shaded region from previous studies known that it's actually slot in or occupies the mRNA entry channel of the ribosome. So that puts the ribosome in sleep. There's no translation going on. We see the same thing, okay, for this region. But also what we see quite interestingly is this, the orange regions are additional region we see on the ribosome. What's striking is this. The N-terminus of SIRBP1 is always anchored to the large subunit. The C-terminus is always anchored to the small subunit. Okay, we can create very nice density fit, and also we did AlphaFold prediction. The computational prediction is actually strikingly precisely matched our model and the density. So we're quite confident about the fitting. So this is actually what we learned. There's some anchoring results, effect of this molecule on the ribosome. What's striking is this. Even in the translating ribosome, where we see a nice mRNA density in the entry channel, this molecule is still anchored on the ribosome. And we have so many states. We actually do some morphing. We can know how actually the head and body domain of the small subunit rotates to let mRNA or this mid region of SIRBP1 in and out of this mRNA binding channel. Basically, we have some model of this, how this as a general regulator of translation always stay on the ribosome in and out of the mRNA entry channel, regulate the needs of translation. But perhaps even more provocatively is this. We see actually this molecule always anchored in the free subunits. This almost gives us a sort of a model like this. In the cell, there's no free subunits. Really means the large or small subunits, even they are not joined together to form the ATS particle, they're still close by, linked by this, so that it will be easy for rapid initiation to deal with translation initiation to deal with some cell stress environments. So we're still testing this model, but that's what we can see from in situ data. Also, we see many other factors. Just to give you one example of this collision factor, EDF1, which is, again, from purified study, the structure has been done. This is a Rachel Green's study published quite a few years ago on the EDF1 in green, in this case, bind to a helix on the ribosome. In situ, what we see is actually binding location very similar, but we see different, say, conformations of the molecule at the termini and also conformation of the ribosome helix, how this molecule sort of hug the ribosome to make a conformational change. If I compare the two and show an animation, you can see the difference very clearly. On the left, you see the animation from purified sample to start, then to what we see in the cell. Basically, it really means if you want to learn the bona fide biology, what's inside cell, of course, is actually the most relevant state. Just an example, okay? And we also can see a lot of polysome. Polysome means actually translation of translation by ribosome, they don't often they are not they don't occur in isolated events. You see a string of ribosome on the same mRNA, they're doing the translation simultaneously. This has been done before, seen before from the low resolution views of the translating samples. Now, in our single particle study, we focused on the middle ribosome, but we see in many different states, we start seeing the neighboring ribosome show up. This is in the wild type cells. When we actually inhibit the cell with the drugs I mentioned, we don't see this. Really means actually during translation, you have polysomes. They may communicate, they may coordinate, they may collide, they may stall, but they always come together. Okay, so we focused on this. Again, we did the classification. We make the much larger single particle box to focus on 2 ribosomes. Did the classification, end up with 2 different classes at quite high resolution, about 3 angstrom resolution, called diribosome structure. Showing a little bit detail here is really you see the 2 classes. They actually chain together by the blue mRNA in the middle, and they have different factors or tRNAs bound in these 2 different states. They have 2 different type of interfaces between the 2, the leading and trailing ADS ribosome. And then if you look at the class 1 on the right, really, if we actually try to build up a third ribosome, according to this type of interface interaction, what we will see is this. After three, two, three are okay. Once you put the fourth one there, you're gonna see clash. They're gonna collide with that. So this is really correspond to what's observed in the field. The ribosomes, polysomes, or disomes or trisomes, 2 or 3 ADS together. On the left, we try to propagate this according to this interface, we can actually build up a helical ribosome. That's also being observed, but the exact function of this different type of polysome is still not very clear. Of course, hopefully our high resolution data will help others to design mutations to figure out what's the function. Then I mentioned we did the inhibitor treatment, quite high resolution, again, not as high as 2.2, but 2.4, not too shabby. We can see very good definition of the inhibitors, and not just atomic models. We can do the translation landscape analysis, look at how the inhibitor actually change the different translation states in cells and compare to its untreated cells. Naturally, because of inhibition, it knocked out quite many translation states. Okay, so I won't go into any details that show you the power of this type of studies. Then finally, just want to give you this is probably my final data slides, just to show that actually besides inhibitors, we can look at a lot of native small molecule cofactors bind to the ribosome. That polyamines in this case, like spermine and spermidine, they are very important for ribosome function. One example, we saw actually a spermidine at the ETRN binding pocket. We see it always there. In the wild type cells, native cells, was the treated cells. But in the inhibitor treated cells, it's part of the binding pocket interacting with the inhibitor. Really, it's part of the inhibition mechanism. This information is actually not entirely new, but it's actually kind of controversial from purified sample. Sometimes people see it if it's included in the buffer. If actually not included in the buffer, you don't see spermidine in there. Really, again, highlight that even for inhibitor design, you want to actually put it in the most native condition that you want to mimic the cell, and of course, doing it inside the cell. If you could do it, that's the most relevant condition. Okay, so sort of summarizes actually really the future is in the cells. Okay, we become a cell biologist. We're trying to figure out different type of complexes in the cell and try to get it to a high resolution. Okay, so what I want to sort of take home message is actually this is sort of a method that bridge the traditional single particle type of work to the cryo-ET type of work you heard from Grant, that it actually has a great potential in situ structural biology. A lot of more need to be optimized. Of course, as Grant already told you that we need better samples to start with. Nothing beats the better physical sample. Get a better milling machine, get a thinner sample, higher contrast, higher throughput, less radiation damage. That would be the best goal to start. Then I give you the example of ribosomes, right? That's the easiest target inside the cell. Of course, most of your target of interest are smaller. Okay, according to theoretical prediction and also a few proof of principle study, the limit of a current 2D template matching is about 1 megadalton. Okay, so for smaller molecules, some larger macromolecular complexes can be done already. But of course, we want to get into the 100, 200 KD realm so that routine structural biology or cell biology can be done inside the cell. There are a lot of ways to do that, to optimize the methods. Actually, start with physical sample to get better data noise, signal to noise ratio is first thing. Now, of course, computation algorithm, how you detect it better using machine learning approaches to detect the smaller molecules. So there's still a long way to go to achieve that, but I think quite hopeful. Then since we have, again, everything inside the cell, we really need better classification or data science tools. We're going to integrate everything in the end, not just this, say, ribosome translation pathways. We're going to integrate multiple pathways in the cell together in the end, right? Then, of course, Grant also mentioned, what if your molecule is very scarce, your target? You only have a very few inside cell. How can you find it, right? So as Grant said, actually, we can use correlated light and electron microscopy to label your molecule to target the region for data collection. But there's another way we're thinking with this, because what we are collecting is a piece of bona fide piece of cell. Really means it has everything. It may not have enough for a single particle reconstruction as of now, but that data set is a data set forever that we can always come back to combine multiple data sets to have enough particle. So that's really calls for a very good database. The entire field need to have a good algorithm, or like EMDB, like PDB, they already started this effort to have a database for the lamellae data, so that everybody can say sort of data mine it for your targeted interest. This is just some perspective. But in the end, really, what I want to emphasize is actually this is not just a standalone technique. What's best is actually combine this with cryo-ET. Cryo-ET, even though it's hard to do, has its actually unique strengths that the single particle way just cannot overcome. Like the third dimension, single particle can define the XY defect direction very well, but the Z height is very poor, really refined by CTF, by large errors. So ET will solve that problem and also give us the sort of the entire picture of the environment of the cell. Sort of the ET give you the picture of the forest, then hopefully combined with single particle, we can get some details of the trees. So really the whole idea is in situ structural biology, both single particle cryo-EM and cryo-ET is really the future of structural biology. And it's actually this revolution is coming up very fast. Okay, so with that, I'll just thank my group for people who did the work and also my collaborators. I think I showed their picture along the way. And I thank you for your attention.
Thank you, Young. Really excellent talk. There's already a couple of questions in chat. Would you like to me to read them out loud, or do you want to read them?
I think I can see them. Okay. So the first is actually by Lee. To my knowledge, in situ single particle analysis requires high resolution templates to locate the targets, to assign the pose or assign the orientation, really means within the cell. It's actually prevent the method to discover the new targets. It is still a barrier, and comparison particle needed for the same projection by single particle and in situ. Okay, so first part of the question is about you need a reference to start with that. Okay, I think it's the comment is partly true. We require relatively high resolution, but relative, it's all relative. High resolution meaning if we get something at about a reference about 8 angstrom resolution, which probably if you think it's an entirely unknown target, 8 angstrom is actually high resolution. But from the traditional point of view, we don't need that high resolution. Okay, but this is actually a bottleneck. So that's actually the one way to solve this is really to combine for entirely new target. Just say there's no reference out there. What can we do? The way to solve this is actually do a quick tomo to actually get some low resolution cryo-ET reconstruction. Low could be actually 10 to 20 angstrom resolution. Then it will help sort of bootstrap the particle picking and orientation in the single particle zero tilt data, so that you can get the resolution higher and higher. So again, these are the frontier of development. I think people started thinking different ways to solve this problem, but it is a problem, but hopefully can soon be addressed. Then the second question is the estimated comparison of particle numbers to needed to compare to purified sample. It does, we don't have an actual numerical number to compare, but really it's all depend on how many high quality particle you can collect. So we could actually get, say, 2 to 3 angstrom resolution ribosome structure with 1,000 ribosome particles. So if you get actually good particles, it's there. But if you cannot collect enough high quality particles, then in general, that is true. You need more particles to achieve similar resolution with a very good purified sample with very good signal to noise contrast level. Okay. Then the next question is actually when you say better milling, you're referring to plasma as opposed to gallium ion and the milling scheme, low voltage and low current. A very good question, actually. The hope is actually we haven't got hand on a plasma miller or especially the new film milling Arctis machine touted by Thermo Fisher, supposed to have low damage. That's actually hopeful. That's true. That's one way to go with the plasma milling, that we can get less damage. Then the lower voltage, lower current is one way to reduce radiation damage. When I say radiation damage, I mean the radiation damage during milling by the particle ions, by the gallium ions. So that's one way to solve it. But that low voltage, low current, the throughput is affected. So there's no good way yet, but hopefully instrument design and algorithm design can help really to reduce this. Okay, then next one is very interesting talk. It is a technique still possible in overexpression system or be overabundant protein lead to increased background. I think the first part is yes, overexpression system certainly will help. When we say overexpression, meaning overexpressed it in cells, really means in this case, I would like to argue the cell is our new test tube. You can still do your mutations, you can still do your overexpressions in cells, but just do it in cells without purify. Overexpression will certainly help. Then you cannot claim that's the native abundance or states or distribution in cells, but still it's in a cellular environment, it's better than your purified sample, right? So it's still very helpful, I think. Then the more protein, I don't think will lead to increased background. The cell is crowded enough. The other sort of unwanted molecule is more of an issue. You have more of your target protein, it's actually a good thing, it's a blessing. Okay, next question. Can you comment on the use of fluorescence imaging for targeting regions of interest? That approach you use a standalone confocal versus in-chamber fluorescence, the wide field microscope. Yeah, we done a little bit work, nowhere near as actually advanced as Grant just mentioned. Try to actually target specific region by combining light and electron microscopy. We done a bit, again, our goal is try to cut corners to make this actually high throughput is our goal. I think to get this generally applicable, throughput is the key. So we could do this actually. We could do room temperature clamp. Really means we could actually express fluorescence tagged targets. Then we actually put it on the cryo-EM grid, look at it before freezing. There, roughly, we know a region that has our target. Then we freeze it. There, we can say correlate to the region by the grid pattern, not as precise as what you already frozen in an inline clamp type of experiment, but still helps. Okay, then next one's with your method. Do you think we can focus on folding and transport of any protein? Of course, any is a big goal. In the end, I hope 5 years down the road, I really hope do. But for now, it's the same question. How much we can see? Do we have the enough particles in there? Do we have the enough size, enough signal to noise ratio? I think this is a moving target. It's getting better and better, but for now, we still have a size and abundance limit. But I hope the limit will move and move. It will be easier and easier. Okay, how can the experiments decide that milling has caused too much damage when too much damage when working on with not so well-known structures? Would a trial and error process be needed? I think, unfortunately, probably yes. But in the end, you will need that if you want to push frontier. But I think there are enough rule of thumb, right, from everybody's experience on damaging. So the Nico, actually, a few groups done damage study. Nico Grigorov's group especially had a published PNS paper on the damaging. In their publications, really, it's actually in the standard milling procedure now. Damage is actually quite severe, meaning up to 60 nanometer from each side of the lamellae actually are damaged. Really means it's a different level of damage. At the very surface of the lamella, damage is probably more severe. Then damage gradually reduce once they go inside the lamella, but still the damage go up to 60 nanometer deep. Really means this is a big challenge. We want a thin lamella to get better contrast. Then if we get to say 100 nanometer, everything's damaged. But I think the way, as we said earlier, maybe plasma milling, low voltage can help this to mitigate the damage issue.
I have late checkout. No problem.
Okay, almost there. Let's say with in situ single particle in cells, do you anticipate that we can get high resolution of plasma membrane proteins, or thin milling still a limitation? Let me see. There are multiple ways if you want to target membrane proteins. Milling, of course, is the most natural, least perturbed ways to get your sample, but there are purified organelles. There are compromised, you can say, break your cells partially, focus on membrane. There are ways to do that. People published method to do that, so that you could do a sort of a combination of half in situ and half purification on that. But this is a very emerging area. I think a lot of bright minds, like people like you guys, can think of how to improve the methods. Okay, then say the recent EMX talk that gallium may be superior to plasma FIB. I think that's Champagne's actually comments. I think we should look into that actually. Really, the jury is really out there. What is the best milling methods? Actually, it's now proven that plasma, xenon type of plasma is better than gallium, sounds like. Yeah, we should all look into this. Okay, then Guobin, thanks for the nice comment. We had a single particle user whose specimen has ice over 100 nanometers thick. They had difficulty in achieving high resolution map. My specimen is over 100 nanometers. The cellular sample give more background than nice. We can get 2-instrument resolution. Do we have any insight into a user's issue on the purified sample? This is a very good comment. Really, this method has no reason, can only be applied to female cells. It is really my belief. This method applied to all the single particle work. We have done actually a few just purified sample. It does perform better. If you have low contrast, your ice is thick. Actually, this method actually can help get to higher resolution. It does. Okay, last question. That's it. Thank you, everyone.
Hi, Yong. It was not clear to me. What do you mean? Why our user had a difficulty to get a higher resolution, but your specimen actually, yeah, you are able to get a better resolution. What's the
Mhm. I think it's this. I think the power of this one is really at the front end of a particle picking and alignment. If you get there, if you do what we typically do is do a local refine. We don't do global refine. The current single particle pipeline, even the most advanced the most recent rely on the cryospark. If you do global refine, the it's just not as good. So the sort of the traditional alignment method, meaning cross correlation based, that's how it actually both in Xinjun and Nico's algorithm, they use cross correlation to align the particle, to identify which is the correct particle and align the poses. Once you've done that, actually, even at a low signal to noise ratio, it actually still performs better than your global refinement, the traditional pipeline. So that works better.
Okay, do you mean in the 3D refinement? 3D
Yes.
3D refinement.
Yes.
So what like a particular 3D refinement do you use? Like a non-uniform or whatever?
Local refinement.
Local refinement.
Yeah. Local
So
only.
you
Since you
think that
started the particle with the orientation already when you pick the particle. That's the key. I think this is the key advantage of this method.
okay, so you think the single particle user the issue was
global refine. We done extensive tests. This is actually not a secret. I think a few very advanced users like Kaijiang's group, they always do this these days, even with the traditional pipeline. Once you're able to define the orientation of the particles, try not to do the global refine anymore. Focus on local refine. Normally, you can get better resolution.
okay, so okay, yeah, maybe we are running out of time, so maybe we can talk later. Yeah.
Okay. Yeah.
Okay, thank you.